or properly controlled can enter into the environment, basically into the fields, which are then taken up by the plants and then will enter into the food chain. Next. The plant used for this study was rice, and these rice plants were grown in a hydroponic system with concentration starting from control with having no nanoparticles, then ranging up to 1000 ppm concentration. Why rice? Because as we have heard in the previous two lectures, it's the cereal grain and the most important food and the second highest worldwide production after maize. So here we have seen that the tabular column shows the AS values, that is quantification of nanoparticles inside the plants, copper oxide nanoparticles. So we have treated these plants for a period of 20 days, and we have analyzed the root and shoot tissues on the basis of 0.1 gram fresh weight, and we have seen that the amount of nanoparticles increases considerably in the shoot as well as the root. So the objectives of my study were first to study the germination rate and the external and internal morphology, the changes, then to study the responses on photosynthesis, and to study the oxidative damage and the protective processes which the plant produces during the effects of these nanoparticles. Next. Okay, in the results we have seen that the germination rate was not much affected. As you can see here, it is almost the same when compared to the other treatments. The root and shoot length was seen to decrease. The root and shoot length was seen to decrease with increasing concentration. Here, this is a pictorial representation of the previous slide. Here you can see in the control plants, the root and shoot length is quite high when compared to the higher treated particles. And these were young seedlings, 6-day-old seedlings. But as you see, once the plants are grown continuously for a period of 20 days, it was observed that the length, the biomass or the length of the roots and shoots were almost similar. They almost came back to the control level, except for 1000 ppm, because there was more yellowing of leaves and because the concentration itself was very high. Here, this might be a result, this yellow coloration was basically due to the decrease in the photosynthetic pigments, and hence it was showing this color. Also, since the plants are able to regain their length as compared to controlled plants, it can also be said that these plants in the further, as the days pass and treated with copper oxide nanoparticles, they could able to mitigate the ROS which is produced by these nanoparticles in the plant, and hence could grow up to a low concentration of 10 ppm.